Hi students, very good morning all. Welcome back to Sri Ramanuja Vidya Mandir. Today I am going to explain about the how do genes control the traits. It is one of the subtopic in our running lesson, that is genetics. So let us start about the how these genes are controlling the traits. Traits means, no children? Trait means character. Okay. First of all, everybody look at the picture. Here cell is there and this is the chromosome and this one is the genetic material, that is nothing but the DNA. Before going to introduce about this genetic material, genes and the DNA, first I would like to explain about the cell. You know that cell is the microscopic unit of every living creature, whether it is a plant, animal or microorganism. Every living creature, in the sense, it should consist of cell. That is the reason cell is named as it is a structural and the functional unit of the living organism. Already we have studied in the postulates of cell theory given by Mr. Stillen and Schwann, right? So in that postulates, the second point is that cell is the structural and the functional unit of the living organism. So as per the guidelines of the some material which is present in the nucleus, this entire cell works. That is the reason nucleus is considered as the brain of the cell. Nucleus is supposed to be present at the center or towards the periphery. It depends upon the type of organism. So in the nucleus, it is the double membrane cell organelle. Basing on the envelope which is present around the nucleus, organisms are categorized into prokaryotes, eukaryotes. I think you have learned about all these things in the previous lesson. Okay, so prokaryote means the cell is the cell in that the nucleus is not surrounded by any envelope. So for example, this is the cell and the genetic material is naked. Okay, there is no certain envelope around this material. So that is the reason we'll call this as a nucleoid, or we can also call it as naked genetic material. So this is the special characteristic feature of eukaryotes. Okay, in the eukaryotes, sorry, this is the prokaryote, right? And in the eukaryotes, the cell having the nucleus with the envelope. So it is having the genetic material inside the nucleus. So that is the major difference between the prokaryote and the eukaryote. Prokaryote is having the naked nucleus or naked genetic material, nucleoid, whereas here nucleus with the envelope and the genetic material is present. Okay, see, this matter means a thread-like structures which are present within the nucleus is nothing but the chromosomal material. See, here a fiber-like structures or network-like structural arrangement we can see when we'll observe this under the electron microscope, somewhat advanced microscope. So we can visualize this presence of some fiber network-like structures within the nucleus. That material is nothing but the chromatin material. Now just with the help of X-ray diffraction studies, Watson and Crick, they gave the structure of DNA as double helical model of DNA. In the previous class we have discussed everything. Okay, now just I'm giving a review of the previous class. This is the chromosome, and if we'll on further visualization we can also see this type of arrangement, means DNA structure. It is the polynucleotide chain. Poly means more than two. Okay, see, nucleotides. DNA strand consists of two polynucleotide chains, means for example this is the chain and polynucleotides will be arranged on this chain, a chain with the beads in the same way. So each circle we can consider as the nucleotide. You know what are those nucleotides? Okay, it's a combination of nitrogenous base and one ribose sugar and other one is phosphate group. Nitrogenous base, ribose, phosphate group is nucleotide. Nitrogenous base and with only phosphate group is known as nucleoside. Already I have explained everything. Here DNA is double helical model of DNA. There are two strands. See, this is the one polynucleotide chain and other one is like this, the second polynucleotide chain. For example, first chain you will consider as a B and the second chain you will consider as a B. So first one A and second one is B. These two chains are antagonist to each other. I mean to say they are complementary to each other. If A strand runs from 5 prime to 3 prime, opposite to this 3 prime to 5 prime. Why it is so? The reason is that, see, here there is a base pairing in between the two strands. I said nitrogenous bases are 4, purines and the pyrimidines. Purines and pyrimidines, right? Purines are 2 whereas pyrimidines are 2. What are those? Adenine and guanine. Next, come to the case of pyrimidines, cytosine and thymine. These 4 are the 4 nitrogenous bases generally we can see in the DNA. Here, one purine is always base pairs with the or pair up with the pyrimidine of opposite strand. For example, so adenine is present on A strand. So opposite to this, it should pairs with the pyrimidines. So that is AT. Adenine is always paired up with the thymine. In the same way, G means guanine. It is always paired up with the what? C. ATGC, cytosine. So it is a compulsory thing. You have to keep it in your mind. Okay, blindly just remember ATGC. A always pairs with the T, whereas G always pairs with the C. And another important point, these two are two separate strands, no? So they need to link together, right? So for the linking, we should use some tying, right? Okay, that tying thing is nothing but the bonds, hydrogen bonds. So A always pairs with the T with the help of double bond, and G always pairs with the C with the help of triple bond. So in this way, entire nucleotide chain is going to be pair up like this. Understand? C, G, again G, C, A with 2, like T. So in this way, all the nitrogenous bases which are present on one chain, it is going to pair up with the opposite nitrogenous base of the second chain. That's the reason I said, so these two chains are complementary to each other. Understand? Next. Let us see this. Here already, see, genes correspond to the region with the DNA, a molecule composed of 4 nucleotides. The sequence of these nucleotides, 4 nucleotides are, just now I explained, okay, adenine, guanine, thymine and cytosine. Like the sequence of these nucleotides contain information about the organism inherit. See students, here this particular sequence of arrangement of nucleotides, it is having the information regarding the trait or a character. For example, my hair color is black in color. So that particular character is present in the form of nucleotides on the genetic material. Not only color, height, about everything. Okay, elastin is the protein which is present on our skin. Keratin is the protein which is present in our hair. Okay, even the nails also have some proteins. Collagen is the protein. So hemoglobin is one of the protein. Okay, so about all this information is present on the genetic material as per this sequence of nucleotides. So the final character, that means phenotype, will be. Okay, understand about a height, about our diseases, about everything, all the information, it is present on the material. That is the reason we'll call this particular material as a genetic material, and this is the only material responsible for the heredity and inheritance. Now you got my point? Right. Now, see, DNA is a double-stranded structure. Each strand acts as a template for creating new partner. In the next slide, I am going to explain this word. And this is the method for making copies of genes that can be inherited. See, Xerox machine, I think you might have an idea. Whatever we'll keep in the machine, same thing we'll be getting. Right? We'll not get something others. In the same way, the genetic material is going to copy it as it is. Okay, that particular process is known as replication and everything. So in the coming slides, I'll explain about. Now, first, let us talk about the genetic code, the sequence of nucleotides in the genes. So for example, see students, here this is the DNA. Okay, so this red one is one nucleotide strand, and whereas yellow one is other nucleotide strand. So it is a double helical model of DNA having two different chains, understand? And the sequence of nucleotide in a gene translated by the cell to produce chain of amino acids creating proteins. Students, I said before, cell, within the cell nucleus is there. In the nucleus, this all process going to happen. Understand? I mean, see, doubling of the DNA means formation of DNA from the DNA process is known as replication. And from DNA to RNA, we'll call it as messenger RNA, mRNA. This particular process is called transcription. In detail, you learn about all these things in the higher classes. Now, mRNA is going to transcribe or translate it into the proteins. Okay, so this particular process is called translation. This is the central dogma of molecular biology: replication, transcription and translation. What do you mean by replication? Synthesis of DNA from DNA. And DNA again going to responsible for the formation of the mRNA. And the technical term which we'll use for this process is transcription. And this mRNA is translated to the protein with the help of translation. See, here, you see this diagram. Here, DNA, RNA. Here, what we can use? Transcription. Okay. And here, from RNA to protein. Translation. Okay, finally, as per the nucleotide sequence, we are going to get the protein. For example, I want the hemoglobin protein. So, in the same way, the protein hemoglobin is synthesized in this way. Not only hemoglobin, all the proteins. Okay, and one more important point. So, for the manufacture of these proteins, the useful organelle is ribosome. Like now, the order of amino acids in the protein corresponds to the order of nucleotides in the gene. The order of amino acids. Actually, what do you mean by amino acids? Protein is a polyamino acid chain. means, so if this is the protein, okay, and this protein is made up of n number of amino acids. Okay, a group of amino acids will link up together to form a complete protein. So for the see how we can build a wall. So by using the bricks, okay, by arranging the grips one upon the other, so we can construct a complete wall. For example, you will consider protein is a wall. The building blocks, okay, the bricks are nothing but the amino acids. Okay, so each circle is nothing but the amino acid. Okay, now you just see this picture, students. As per this coding of nucleotide, so we are going to, there is a chance of formation of amino acids in the protein. So it is completely basing on the information which is present in the genetic material. That is the second point. The order of amino acid in the protein corresponds to the order of nucleotide in gene. This relationship between the nucleotide and amino acid is called genetic code. Now you understand, students, what do you mean by genetic code? The relationship between the nucleotide on the DNA and the amino acids on the protein is genetic code. Now, the amino acids in the protein determines how it folds into three-dimensional shape. This structure is in turn responsible for the protein's functions. Okay, so this is also not only responsible for the synthesis, and it's also responsible for the functions of the particular protein. Right. Protein carry out almost all the functions needed for cell to live. Understand this point, students. So we as a living organism, we do require help of some proteins always in our day-to-day life. Okay, a change in the DNA can change the protein amino acids, thus changing the shape and function. So this can have the serious effect in the cell and on the organism as a whole. So people will suffer with the sickle cell anemia. Sickle cell anemia, it is one of the disease, students. Sickle cell anemia, which is caused due to the change in the single nucleotide. Even the small change also, it alters the particular character which is exhibited. So this is about the code. Next, very important topic, that is sex determination. Let us discuss what you mean by sex determination. Sex determination is a biological system that determines the development of sexual characters in organism. So it determines the development of sexual characters. Sexual characters in organism. Right? Let us study in detail. Most sexual organisms have two sexes. What are those? Male and female. Okay. Genetically, both the sexes have different alleles or even different genes that specify their sexual morphology. So, as they're having the different allele or different genes for the sex, so it is giving the different morphology. And in human, the total chromosome complements are, so when we'll talk about the humans, so the total chromosomal complements are 46 chromosomes are present. Okay, out of 46, 44 are known as autosomes, whereas rest of 2 is known as allosomes. And it is also known as sex chromosomes. so which determines the sex of an organism and various sex linked characteristics. So not only it not only determines the complete sex of the organism, and it also determines certain characteristics of sexual characteristics. Okay, in most animals, those that have the XX chromosomes are female, so female always used to have this condition XX, whereas in the male XY is there. Okay, this is the standard chromosomal conditions of allosome in the human. XY means male, whereas XX means female. Now, in humans, sex is predetermined in the sperm gamete. You know that, students? See, there is a, what do you mean by sexual reproduction? So, fusion of male gamete with the female gamete, right? Okay, what is this male gamete? It's nothing but the sperm cell. Sperm cell. And here in this female, it is egg. Okay, these two gametes having the condition of sperm cell is having the condition of XY, whereas egg cell is always having the condition of XX. And we'll call this as a homogametic. The reason is that here the both are X, right? This condition is called homogametic, whereas this we'll call it as heterogametic. Okay, now see here, because in the sperm cell we have two different things, X and Y, that is heterogametic, and in the case of egg cell only two X's are there, so that is homogametic. Hence, there are two possibilities that can occur during the fertilization between the male and the female gamete, that is XX and XY. Since sperms are variable factor, so the sperm is the variable factor. He is the man only, the male only responsible for the sex determination of the child or a baby. So the one carrying X chromosome or the one carrying Y chromosome, it depends upon the sex of the baby is completely depends upon the male only. Understand? Right? Diagrammatic representation, students. So male is having the XY condition, whereas female is having the XX. See, gametes. If the gametes are formed, so this is the sperm cell, and one sperm cell may have the X information and other sperm cell may have the Y information. Okay, Y. And other one is having the Y. So here in the case of only one egg is there, so only X condition is there. Right? In the offspring, we can expect if the zygote is with it mutes, if X is going to combine with the X, so XS will be, if the Y is going to combine with the Y, so we'll get the male child. So in this way, the probability of sex in the progeny. Let us see the sex determination in the human beings. Father, parent, is having the condition of XY, whereas mother is having the condition of XX. See, when the gametes, okay, X, Y from father, XX from the mother, okay, and if this X is going to combine with this X, we'll be getting female. Okay, in the next case, also female. Okay, 2XX conditions. Okay, here in this, both things, male and male. So finally, in the offspring, 50% probability of a female child and 50% probability of male child. Understand? See, the Punnett, if you'll check the checkerboard method, so in this way, see, this is the egg. So 22 is nothing but the pair of chromosomes, that is autosomes, and these are the allosomes or sex chromosomes. So this is the Punnett diagram or checkerboard method. And let us see few more information about this. Many sex discrimination systems exist in a number of insects including grasshopper, cockroach and the crickets. So there is a variation where female have 2 copies of sex chromosomes but male have only one, that is X. So here the 0 indicates or O indicates the absence of the second chromosome. So in the grasshoppers and the cockroaches and the some other insects, we can see the X and Y, X and O in the male. Okay, XX in female. Okay, now in some species of reptiles like turtles, the sex determined by temperature at which egg is incubated. In tropical clownfish, the dominant individual in a group become female while others are male. So it is depends upon the organism. Next, very important point to be noted here. Some species have no sex determination. No sex determination. Some species have no sex determination system. So what are those species? Certain species of example snail and earthworms. Snails and earthworm are considered as the hermaphrodites. Considered as hermaphrodite. Okay. So here there is no sex determination system. A few species of insect, fishes, reptiles reproduce by parthenogenesis and all are females only. It's a particular species such as insects, fishes, reptiles, they'll produce by means of parthenogenesis, all are females only. This is about the sex determination. And another important topic, that is karyotype. Let us study about the karyotype. The chromosome complement of a cell, the entire chromosome complement of individual or a cell. Okay, so a karyotype includes detailed morphological structures of each chromosome of a set. In man, 46 are there, I said, no. See, 2n is equal to 46. Okay, in the humanities and in the onion, the condition is 16. 2n is 16. The chromosomes are found therefore in their karyotype is 23 and 8 pairs of homologous chromosomes are present respectively. So this is about the karyotype. Now I am going to explain about the ideogram. Ideogram. So a ideogram depicting the arrangement of homologous chromosomes in the decreasing order of their sizes. So all these are the 23 pairs of chromosomes. They were arranged basing on their sizes. So this particular arrangement or basing on the sizes is known as ideogram, or a pictorial depiction of the karyotype is called ideogram. So a diagram depicting the arrangement of homologous chromosomes means, so a pair of chromosome is called, this is the homologous chromosome. Homologous chromosome. Okay, this arrangement of this homologous chromosomes as per their size, that is called ideogram. Now let us see about the evolution. See, evolution is the product of two opposite forces. Okay, so the process that constantly introduce variations in the trait and the process that make particular variant become more common or rare. Okay, the main cause for the variation is mutation. Very important. mutation. So a gene flow, a reshuffling of the gene in the sexual reproduction which changes a sequence of change. So there is a change in the sequence of gene. What is a mutation means? Change in the sequence of gene. Right? Now, see, the two main processes which cause variant to become more common or rare in a population or natural selections which cause traits that help survival, reproduction to become more common, and traits that are hinder the survival, reproduction to become more rare. Natural selections occur. See, so this is about the, now in this lesson we are going to learn about the evolution also. Okay, evolution is the product of two offspring or two opposing forces actually. The process that constantly introduces the variations means mutation. What do you mean by mutation? A change in the genetic sequence is called mutation. I said no. Here, in the previous slides, I have explained about the nucleotide sequence. If any change in any particular order or nucleotide sequence, that may be represented as the mutation. So, that particular character may alter the character in the progeny. Understand? This is about the evolution. And now let us see the next one. Mutation means a change in the genetic sequence. What do you mean by mutation, students? A change in the genetic sequence. There are many different types of mutations causes. Okay, some mutations are harmful while some other can be beneficial. So there are some variations, they are harmful, and there are some variations, some are beneficial. Right. Evolution and the natural selection. So students, natural selection occurs because only a few individuals in each generation will survive. So students, as per generations continues, only a few individuals can survive, whereas remaining all dying. What is the reason? Simply by the principle of natural selection. Since the resources are limited and organisms produce more offspring than their environment can support. So here, the organism is able to produce more number of organisms. So this much number is unable to support it by the nature and the environment. Over many retained. So this causes adjustment of traits so that they become suited to the organism's environment. These adjustments are called adaptations. However, not every trait is adaptation. See, actually, as the days going on, years are going on, there are new climatic conditions are there. Adverse conditions we need to face. So the individual or the organism which can have the efficiency to adopt those adverse conditions, only that can only survive, whereas which cannot adopt, simply they are dying. So this is nothing but the process of natural selection. So which have the more power, more resistance power, more capability can only survive at last, whereas if they have less power, less resistant power, less capability, everything, they are going to die. We need to compete with the adverse conditions shown by the environment. So nature only allowing us to survive in the nature. So if nature it will not support us, we cannot survive. Right? Next. So that is the adoption and adaptations. Evolution, a gradual development of more complex species from pre-existing simpler forms, is known as evolution. What is the evolution definition, students? A gradual development of more complex species, okay, from pre-existing simple form. Actually, our previous ones are we'll call them as primitive. Okay, so the pre-existing is the development of more complex species. Okay, the gradual development of more complex species from pre-existing forms is known as evolution. Understand? Next, actually some theories were given in the evolution. A number of theories have been put forward to explain the mechanism of evolution of animals and the plants. Okay, some important theories are, first, let us talk about the Lamarckism. What is that? Lamarckism. Okay, he is Mr. Jean Baptiste Lamarck. Jean Baptiste Lamarck tried to explain the principle behind the process of evolution in a book called Philosophy Zoology. Okay, Lamarck proposed the theory of inheritance of acquired characters. So what he gave, students, he proposed the theory of inheritance of acquired characters. Inheritance of acquired characters. He proposed a theory that is inheritance of acquired characters. According to this theory, so there are some points. Let us discuss those points now. All living things and their components parts are continually increased due to the internal vital forces. All the living things and their component parts, they are continually increasing to the internal vital forces due to the. And let us see the B. Environment keeps on changing and thus influence the organism by creating new needs. The new needs are met with by producing new structures. The changed environment puts certain old organs out of use. Okay, so very important point. Environment keeps on changing and thus influence the organism by creating new needs. So here, because of the environment only, so here the organism is creating a new needs. These new needs are met with by producing new structures. The changed environment puts certain old organs out of use. When new ones are coming, so old ones are, it is out of use. A constant use of organ results in better development, whereas disuse leads to the ultimate degeneration and the loss. So the modification produced during the whole life of an organism are transferred to its offspring. As the process is continued, after several millennia, a new form will be formed. So these are the different things which we can see in the case of evolution. It is given by the Mr. Lamarck. So he gave the theory of inheritance of acquired character, theories of inheritance of acquired character, and in the book Philosophy Zoology, so he gave one theory. Actually his full name is Mr. Jean Baptiste Lamarck, and try to explain about the principles of behind the process of evolution. Okay, next, and so that's all for today's lesson, students. In the today's lesson, I gave you about explanation about the how do the genes are able to controlling the traits or a character in the individual. The main responsible factor I have explained, and I have explained also about the genetic sequence or genetic coding, and next how this genetic information is going to transform in the form of a protein or translate into the form of protein. I have explained that also, and another important point about sex determination. So, and sex determination is a biological system that determines the development of sexual characters in organism. So here in the human we have 43 chromosomes. Out of 43, 44 are autosomes, whereas remaining 2 are allosomes. Allosomes are also known as sex chromosomes. Okay, see, the male one is having the XY chromosome, whereas female one is having the XX chromosomal condition, right? And in the next lesson, next slide, so male condition is called heterogametic condition, whereas females are in homogametic condition. And this is the diagrammatic representation of sex determination in the case of human. Next, here the father and the mother, how they'll combine, how the probability of the female child and the probability of male child we have discussed here. And see, this is the Punnett square or checkerboard method. The sex determination is also described in the other organisms like grasshopper, cockroach and all. Here the male is having the XO condition. Okay. And there are some other organisms, they are not having the proper sex determination system. So example like snail and earthworms, will call them as hermaphrodites, but there are some insects, fishes and all produced by means of parthenogenesis. Very important, parthenogenesis. Understand? So that's all for today all. Thank you so much. Thank you all.